Hello, this is Catherine ASMR, and today I'm going to be talking to you about one of my favourite things, and it's going to be quite tricky for me to keep my voice even and level, because once I start talking about this I'll probably end up trying to just say as much information as I can. So what I'm going to be talking about is photosynthesis, and I'll be going into quite a lot of depth, but I am going to try and explain everything as simply and as fully as possible so that you can get the gist of what's going on. I may still confuse everyone, but so when I was testing out my shot earlier, I realised my whiteboard was way too big for the shot, so instead I'll be using a marker and pen. So photosynthesis can be split simply into two sets of reactions, the light dependent and the light independent reactions, so called because the light dependent reactions are dependent on light and the light independent reactions are not, at least not directly. So light is a big part of the light reactions, so that's where we'll start because it's the first set of reactions to happen in photosynthesis, which is essentially turning carbon dioxide into sugars, which is the basis of all life, really. If plants didn't do this, we wouldn't be here, so it's pretty important and pretty exciting as well, because plants are one of the world's biggest sinks for carbon dioxide, which means that they are actively slowing down climate change because they're absorbing that much CO2 from the atmosphere, which is what warms our climate up. So we owe them a lot. So here is a leaf. You'll have to use your imagination with me. So that, for all intents and purposes, is a leaf. And inside the leaf are chloroplasts. These things. So where I've drawn maybe 20 or 30 there are actually, and I'll have to double check this, and if you see a little note in this video that means that I have messed up slightly, but I'm pretty sure there is 500,000 roughly chloroplasts in each square millimetre of a leaf. I could be wrong, I could be very wrong, but it's definitely 0.5 something. It's definitely 0.5 million in a certain square meterage of the leaf, and I do think it is millimetres. So these chloroplasts are where the light dependent reactions take place, and chloroplasts, these chlorophyll pigments within the chloroplasts, they're arranged in stacks like this. So imagine that's the leaf, but this time, say that's the top of the leaf. Now we're looking at a transverse section. So imagine that I've cut open a cake and you're looking at all of the fillings of the cake. That's the equivalent of what's happening here. So these chlorophyll pigments are stacked in things called grana, which are called their thylakoid membranes, and the stacks are grana. So these are all chlorophyll, well, they're all photosynthetic pigments, and this one at the top, these are called antenna pigments, and it's pretty self-explanatory what they do. is these are the photosynthetic pigments that absorb solar energy, that is the light energy from the sun, and that's what starts off this whole thing, the total the foundation of life. So these antenna pigments, they absorb light, and this light excites some electrons in the photosynthetic pigments, and as it does so, these electrons are shot to a higher energy state, which we call excited. So these electrons are excited, and as they return to a stable state, they release energy as both heat and fluorescence. So imagine them just shooting up, and as they come down, they're releasing heat and they're releasing a red-orange fluorescence, and this fluorescence, this light energy, is passed from photosynthetic pigment to photosynthetic pigment. And we call this method of energy transfer, resonance energy transfer. So this is happening in photosystem 2, and they don't happen second. It's just the fact that photosystem 1, which happens later on, was discovered first, and they haven't bothered to update the names of these things, even though it's ridiculous. So photosystem 2, everything happens first. Photosystem 1, everything happens second, which is ridiculous. But okay, so this is photosystem 2, and so it travels via resonance energy transfer through the pigments until it reaches, so say, and then each photosystem has a reaction centre. This is the reaction centre, and they're also photosynthetic pigments, and these are called the P60 molecules, and these are the pigments at the centre of the reaction complex. And this is kind of the heart of what happens at photosystem 2, because once it's reached the reaction complex, this energy causes an electron to be passed via what we call an electron transport chain, which is just essentially, imagine it as a little journey that the electrons have to go to get to photosystem 1. So they go down the electron transport chain to photosystem 1, and these molecules also accept an electron from water. This is because when it loses an electron, because electrons are negatively charged, so imagine the electron like this, and that's being lost from the P60 molecules. So the P60 molecules are now positively charged, which means they want to return to that stable kind of state. So it accepts an electron from water, and that causes water to be split with oxygen as a byproduct, and protons are also generated, H+. And these are important, but I'll explain a bit later. ATP is also generated at this stage because part of this electron transport chain has a proton pump. So when you have protons like this and you create like a gradient of protons, there's a complex that's part of this stage that pumps the protons, and that works as an energy source to generate ATP, which is the basic unit of energy we all use in our cells to generate any kind of energy. So if you think it's glucose or it's a carbohydrate and that's our basic form of energy, it's actually not. That's not what we use in our cells. That provides the energy. It just enables that form of energy to be produced, essentially. So on to photosystem 1. Then after the electron transport chain, in PS1, essentially the same thing happens. So actually I didn't need to turn over that page. So imagine it's very similar. It's a stack of photosynthetic pigments with a reaction centre at the middle. And the only real difference between photosystem 2 and photosystem 1 is that these molecules, called the P60 molecules, they're called the P680, they're called the P700 molecules in photosystem 1. And this is just because their favourite wavelength of light to absorb, or the wavelength of light that they're optimised to absorb, is 680 nanometers and then 700 nanometers. So I'm pretty sure that that's the violet end of the light spectrum. So their favourite bits of light energy to absorb are at the violet end. That is more violet, so it gets more of the solar energy at the far end of the light spectrum, and this will get more closer to the red wavelength of light on the light spectrum. And actually, it might be better just drawing this out. So I don't know if I'll get this the right way around, but basically they absorb light up here and up here, and it's the same. So these are the two pigments, so they're just better at absorbing different parts of the light spectrum, and this just means that they can use as much energy as they possibly can. And actually I should have drawn this line down here, and here is the green wavelength of light, and actually they don't absorb it, which is why chlorophyll, and by extension plants, are green, because instead of absorbing the solar energy, the green solar energy for photosynthesis, they reflect it back. So that is why plants appear green. Okay, so photosystem 1, same things happens. We're still in the light dependent reactions now. So yes, that concludes the light dependent reactions. And also here, so we've generated energy along the electron transport chain in the form of ATP, which is essential to what we call fix carbon dioxide into a carbohydrate. So yes, this ATP here, and then in photosystem 1 it's passed to, actually I'm not going to write it out because it'll take me too long, but it's passed to an enzyme called NADP plus reductase, and what that does is transforms NADP so we've got these NADP plus molecules hanging around, and you know how I said that when something's positive or negative it prefers to come to that state, or it's happy to come back to a neutral state. So what the NADP plus reductase does is it allows that to be turned into NADPH. So what that NADPH does is that it provides reducing power, and what that basically means is it can donate an electron to whoever it likes, and this comes in handy later. So yes, we've got ATP for energy and NADPH providing reducing power out of the light reactions, and they're going to help us in the light independent reactions. So that's where we're going next. So with the Krebs cycle, it basically has 3 stages, and that is carbon fixation, and this is where carbon dioxide it bonds to a 5 carbon compound compound called ribulose 1,5-bisphosphate. The reason it has the numbers is just to do with where the phosphates are in terms of which carbon, because the carbons are all labelled 1 to 5 if there's 5. I don't know if that was a necessary explanation. So the carbon binds to this 5-carbon compound, forming a very temporary 6-carbon compound, and then that splits into 3-PGA. So 2 molecules of 3-PGA, and this first reaction, which is vital, is catalysed by an enzyme called rubisco. And rubisco is thought to be the most abundant protein and certainly the most abundant enzyme in the world. That could be proven wrong eventually with science, who knows, but for now that's what we think. So incredibly important. It allows the first, it allows carbon to be turned into a carbohydrate of sorts. So carbon dioxide plus ribulose 1,5-bisphosphate 5-carbon compound turns into temporary 6-carbon compound, which then splits into 2 3-carbon carbohydrates, which in turn turns into 2 3-carbon simple sugar type carbohydrates. And the next step is reduction. So this is where our NADPH comes in handy, because they can reduce, they can use their extra H to reduce another molecule by donating its electron. And that's how we transform the 3-PGA molecules into the 2 3-carbon simple sugars that we get here. And then, because it's a cycle, and if it's not a cycle, we'd all have died very quickly if we were only around for 1 ton of this thing. Because it's a cycle, the first 5-carbon compound, which was ribulose bisphosphate. Won't worry about the carbon numbers right now. So this is essential, because then it can go on to do more carbon fixation, because these ribulose bisphosphate molecules are essentially just a carbon dioxide acceptor. That is its purpose. It's what allows these carbon dioxide to turn into this 6-carbon compound by joining with this 5-carbon compound. So if you'll notice, since this is regenerated, this 5-carbon compound, there is only a net total of a 1-carbon sugar created from this cycle. So actually, to get 1 6-carbon carbohydrate, we need to do 6 turns of the cycle, which seems like a lot of effort, which is probably why there's so much rubisco. Trying to think, is there anything else I've missed? And also this 3-GPA molecule, more than just the reducing power, supplied by NADPH, which donates its electron, ATP that we also got from the light reactions, is also used here, because the enzymes that turn this 3-PGA molecule into this simple sugar molecule requires ATP, in other words energy, to be able to form that. So that's where they come in handy, and then they go on and are regenerated into ribulose bisphosphate and 1 simple sugar, 1 carbon molecule. So 6 turns and you have yourself a full carbohydrate. So we've taken light energy from the light reactions, and we have turned carbon dioxide into a carbohydrate with the light independent reactions. And that is the foundation of life. That's how we're all able to live, and something that humans cannot yet do, though hopefully maybe in the future. Also, I realised that I might have done this, I'm not sure if I have, but I may have called it the Krebs cycle at one point, but I actually meant the Kelvin cycle. The Krebs cycle is very different. So yes, that was the light independent reactions are also called the Kelvin cycle. So yeah, there we have it, the light dependent and the light independent reactions, and how they take light energy from the sun and use that to drive the fixation of carbon dioxide, using lots of other, intermediate helpers to turn carbon dioxide into a 6-carbon carbohydrate that provides us the energy storage molecule that we all use. And without those plants, we would not be here today. So I hope you enjoyed, I hope you followed. If you have any questions or want to point out that I've made an error, please go for it. I'm not perfect, but I think I know the subject reasonably well. So I did my best, and hopefully you learned something new. But otherwise, I will see you in the next one for slightly less intense content.